Good afternoon, students. Yes, Itanagar, Shillong, Tamilong, Chuachandpur. How are you? Yes, Shillong, please share your camera port number. I am not able to see anybody. Yes, how are you? Children, are we connected? Yes, please let me know if we are connected. Good afternoon, Tamilong. So, how was your test? You had a test? Did you have a test on Saturday, Sunday? Yes, tell me. Okay, fine. Yes, Shillong. Saturday, so how was it? How was your test? Yes, centers. Tell me, how was your test? Okay. Good afternoon, Senapati. How are you people? Why not good? What happened? Why it was not good? Why it was not good? Tamilong, tell me. Why it was not good? Yes. What happened? What was the issue? Okay. And how was the set? How was the test? Okay, 9 present in one class. Yes. Okay, so are we supposed to discuss something? Do you want to discuss any kind of doubt, any kind of problem? Anybody? Good afternoon, Shillong. Yes. You wish to ask any doubt, any problem, or shall we start with a new chapter? Isn't it? We are supposedly going to start today a new chapter. I told you earlier also. Yes, camera port number 8084, isn't it? I guess this is not the class I am looking at. I guess that. Okay, fine. So let me start the new chapter. Okay, podocytes are the particular cells. I taught you podocytes. When I was talking about glomerular filtration, I must have told you about podocytes. There were these special structures like this. Remember glomerular filtration? So there was a particular structure like this when I was teaching you that. And there are special cells on the Bowman's capsule, and these cells, they are called as podocytes. So this I have taught you, I remember. This kind of structure was present, and I must have told you this is capillary pore. This is having a size of 10 nanometer. This is podocyte, and in between this we have a structure called as filtration slit, supposedly of 25 nanometer, and this is the capillary of the glomerulus. This was basement membrane, and this I must have told you was Bowman's gap. So just go across your notes, just go through your notes, you will find that typical structure called as podocytes. So these are podocytes. Podo means foot, okay, and cytes means cells. So these are specialized cells of foot-like structure, just like food processes. Okay, clear? Fine. So what was next thing, ma'am? If thyroid does not function well, can be, but however we need to do biopsy and then we can conclude. Functioning, over functioning is obviously can be cancer. Under functioning needs a better throughout into it. So we cannot just conclude that if an organ is not working properly, we cannot conclude cancer exactly. So we need to go for biopsy. We need to see the cells, we need to see the particular kind of expressions, kind of proteins present. Then only we can conclude about cancer. Yes, any more questions? Yes, Shillong. Senapati, Tamilong. Okay, fine. So did you get podocytes in my notes that I taught you? You must have. So let us start a new chapter. So we are done with the whole unit of human physiology, and we will be starting with a new chapter, that is chapter number 4 of your book, called as Animal Kingdom. Okay, so this is important chapter in terms of like questions that directly come from this chapter, and the examples of different kinds of phyla, they are moreover more important. So you need to learn the examples, you need to learn the particular classifications, and or whatsoever is given you in a book, you need to learn that, because questions will be coming directly from your book without any kind of modification, without any kind of changes. I will be giving you some questions from the previous year NEET papers also, so that we can get an idea that how the questions are being asked from Animal Kingdom. So before we start this chapter, let me know if we have done with biological classification and living world. Are we done with biological classification and living world? Just let me know. Since this is a chapter with continuity of that, in continuity of that unit only. So did we do? Have we done that chapter, biological classification? Okay, fine. Living world has been done. So what is the particular taxonomy? What is taxonomy? Answer me some questions. What is taxonomy? Answer me. What is taxonomy? Yes. What is taxonomy? Yes, children, answer me please. What is taxonomy? Living world has been completed, so you must be aware about taxonomy. Tell me. Yes, different species, is it? Okay, anybody else? Any of the centers? Tamilong, Senapati, Itanagar. Yes, Itanagar is connected. Churachandpur is connected. Yes. Tell me, what is taxonomy? The study of different taxon. And what is taxon? See, when you talk about taxon, you talk about kingdom, you talk about phylum. Sir must have told you the word phylum is not used in plants, and we use a word called as division. Yes. Then we have class, followed by order. Then we have family, followed by genus, and finally we have species. So we have these many things, and they are all taxon, like kingdom is a taxon, phylum is a taxon, class is a taxon, Similarly, order, family, genus, species, all the components are a taxon. Taxon is singular, taxa is plural. So, when you talk about these particular things, we talk about taxonomy. So, according to Whittaker, we have divided the whole of the living world into 5 basic kingdoms. What are they? Answer me, what are the 5 basic kingdoms? Okay, directly. Okay, fine. Very good. Yes, I want answers from all the centers. Everybody was attending those classes. You cannot forget these things. This is very, very basic. Yes, very good, Shillong. And what about other centers? Yes, Senapati, Itanagar, Tamilong, tell me. The 5 basic kingdoms according to Whittaker. Whittaker divided the whole of the living world into 5 basic kingdoms. Name those kingdoms. Quickly. So, we have a mnemonic to learn the kingdom, phylum, division, class, order, family, genus, species in a sequence. You can learn it using keep pot clean or family gets sick. Sir must have told you something else also, but you can use this also, or either ways you are comfortable. So, in this way you can learn all the particular taxon in a sequence without any, because many times in NEET they ask you this sequence also. So, it is up to your luck. If you are lucky, you will get these kinds of questions. Okay, fine. So, we have divided it into 5 kingdoms, isn't it? R.H. Whittaker, year. Who is going to tell me about the year? Is it 1969? He divided all the particular members of the biological world into 5 kingdoms: Monera, Protista, fungi, Plantae, and Animalia. So, I must have taught you all this. I am going to teach you this thing, Animalia. So, Animalia is all about animals, isn't it? Animalia is all about animals. And how do you categorize animals? Because when you talk about Protista, you will find in Protista a variety of animals, variety of organisms, sorry. You will find in Protista a variety of organisms. Like if you talk about a Protista, the first 2 classes of the Protista, they are actually photosynthetic. The chrysophytes and the dinoflagellates, or diatoms or the dinoflagellates, they are photosynthetic, first 2. When you talk about the next phyla, it is basically holozoic as well as photosynthetic. Or you talk about some like a phyla which is fungi like also, better to say it is saprophytic. If you are talking about mode of nutrition, we would be saying it is saprophytic. That was slime mounds. Then you talk about a particular phyla which is holozoic, you have 2 under them. One is having both photo plus holozoic mode of nutrition, and the other is only having holozoic. That is the particular group called as protozoa. So, you have protozoa and you have euglenoids. So, we are considering the protozoans as a particular group of organisms that are thought to be the primitive animals. So, it is according to many evolutionary biologists, people say that protozoa was the organism which was primitive, or which is primitive animalcule. Animalcule means small animals. So, these are the small animals, these are the small microorganisms Exactly, microorganisms, because we need a microscope to see them. And what kind of members are placed in protista? If you talk about protistans, all of them are eukaryotic, plus they are single celled. So any of the organism, if it is eukaryotic and possesses a single cell, they were placed under protista. Okay. So in saprophytic we are having slime molds. So who is going to tell me that why we did not place such slime molds under kingdom fungi? You have done with all these things. Tell me why we don't place the slime molds under kingdom protista. Yes, quick. Don't have much time. Yes. Quickly start giving answers, all of the centers. So protozoa is about what? Protozoa is about number 1, eukaryote, number 2, they are single celled, and number 3, all of them don't have a cell wall. They might be having a protein covering over them called as pellicle, but they don't have a cell wall. Now, my question was, why have we put slime molds over here? Look over here, everybody. Why have we put slime molds under kingdom protista? Why don't we put it under classification of fungi itself? It also happens to be a fungi. So why not to put under fungi kingdom itself? Yes. Please answer. You have done this chapter, you told me. Half of the people are sleeping, I can see at Shillong. Yes. Get up Shillong, I know. It's very nice weather, post lunch session. All the effects are cumulative. Plus animal kingdom classes. Yes, quickly answer me. I am waiting. Yes, children. You told me you have done this chapter. Shillong, Itanagar, Senapati. Yes, children, you need to answer this question, please. Children, please answer this question. Yes. Do you know this answer? Do you know its answer? Yes. Tell me. You have. Why don't you put slime molds under kingdom fungi? Why did you create a different protista under division for it? It's called as slime molds. Why? Yes. Should not have taken so long. Exactly. Very good, Shillong. It is lack of chitinous cell wall. Cell wall is not sufficient. You have to mention what? You have to mention that slime molds, they do not have cell wall. Yes. And the cell wall should be what? Of what? It should be always and always made up of chitin. Because of this reason, we have not put slime molds under kingdom fungi, because all the members of kingdom fungi, they are having cell walls. Okay, being unicellular will be a wrong answer, because there is an organism called as yeast, which again is the only exception for unicellular fungi. So if you talk about unicellularity, it will be wrong. So you have to mention this point if a question comes to you in NEET, that you have placed slime molds over there, because why? The reason being that they do not have cell wall made up of chitin. Very specifically. Okay, very specifically you have to tell that the cell wall is not made up of chitin. That's all. And what about dinoflagellates and diatoms? Why did not you put it under plantae? Yes. Why have you not put diatoms and dinoflagellates under plantae kingdom? They are equally photosynthetic. In fact, diatoms, they are the largest producers of the ocean. This is the question always coming. So remember this thing. If you talk about diatoms, They are the largest producers of the ocean. Okay, just mention these points. These are the particular things where they are asking questions regularly, on a regular basis. That what are diatoms? So diatoms, they are the largest producers of the ocean. Okay, come back. Okay, so this is the reason why we do not put, have not put slime molds under saprophyte, under fungi kingdom. So talking about again protozoa, protozoa has three characteristic features always, exactly same reason, very good. So we have eukaryotic, they are single celled and they are without cell wall. Is it, Shillong, they don't have a cell wall? Are you sure about it? Because as far as I remember, diatoms, they have a cell wall of silica. The cell wall is made up of silica. And we talk about dinoflagellates. Their cell wall is made up of cellulosic plates. Just cross check these things. You should be ready with the correct answer in the next class. The cell wall is made up of silica, whereas in this case the cell wall is made up of cellulosic plates. Okay, so come back to protozoa. So protozoa is said to be the primitive animalcule. So they are having many characteristics that an animal possesses, that an animal has. But yes, they are single celled and none of the animals are single celled. So what we did is that we created a different class or division under the kingdom Protista and we placed this particular division over there only, because in animals we are having a typical kind of features. The features of kingdom Animalia are, all the animals, they are eukaryotes and they are, yes, they are multicellular. Two most important characteristic features of the animals that makes them, and obviously lack of cell wall would be the next important character. So they are what are animals? Animals are eukaryotes, animals are multicellular, animals, they don't have a cell wall. So with these properties we ought to learn about the basic differences. Always keep in mind that you should know the basics should be clear. Why we are doing a particular classification, it should be clear to you always. Why is this thing made under like this? They are having saprophytic like of conditions, nutrition, but why we have put it under Protista? So you should be able to think these answers, isn't it? Why is bacteria, which is photosynthetic, not placed in a plantae? Yes, because bacteria is a particular kingdom, or Monera is a particular kingdom that is solely made up of prokaryotes. So if it is prokaryote, it needs to be placed in a separate kingdom, and that kingdom is Monera, isn't it? So you need to know the basic particular points of division. Why Whittaker did these things? You should be able to know these things, because they are often asking questions from the basics. Okay, so basic should be clear. So everything is clear up to this. Rest everything you can just go back home and sit and think about the points. Yes, tell me if these things are clear. Everybody. Yes, children. Are these things clear? Everybody, Itanagar, Shillong, all of you. Shillong, things are clear? Senapati? Okay. Fine. Yes, that is a particular homework I gave to you. You have to go and find what makes it unique. Why do you not find flagellates? You should think these things, isn't it? Just go back home and find it out. Otherwise, I will be telling you tomorrow. No issues. I will answer all the questions tomorrow. But But yes, if you find out on your own, you will be able to get new things also. So along with that, you will find 10 more new things. Okay, fine. So try today. If not, I will answer you tomorrow. Okay. So let us proceed ahead with the chapter that is typically animal kingdom. Now let's start with the main chapter. So animals, they are also present in varieties, isn't it? We have lots of animals placed under the animal kingdom. When talk about animals, we have lots of animals, isn't it? We have many animals. So there is a need to classify. Did you copy everything on the from the previous slides? All the centers. Did you copy everything from the previous slides? Please let me know if you are out to note anything. Did you copy everything? Centers, let me know please. Itanagar, Shillong, Senapati, Tamilnong. Did you copy everything? Tell me. Must have. Okay, let's start up with animal kingdom. So what are the members of animal kingdom? Who are the members of animal kingdom? Just now we did that. Everybody who is eukaryote, everybody which is multicellular. So everybody which is eukaryote, multicellular, do not have cell wall, isn't it? And obviously they are holozoic, or what we can say is heterotrophs. So heterotrophs means those animals which are depending on others for food. And holozoic means what? Holozoic means those animals which consume the food as such, that is in complex form, and break down it inside their body. So complex to simple is carried out in their body. So you will never call a fungi to be a holozoic, having a holozoic mode of nutrition. You will never call a parasite to have holozoic mode of nutrition. But yes, we are having, a rat is having, cow is having, a goat is having the simple breakdown from complex to simple inside its body. So what we call them, these animals to be, we call these animals to be possessing holozoic nutrition. So this is what is called as holozoic nutrition. Clear? All of you? Any problem up to this? Let me know please if any problem. It's clear? Just copy and let me know if you have if that has been done. You can just reply me back on the WhatsApp if your chats are not working. I'm not able to get many of the centers. If your chat is not working, you can just reply me back on the WhatsApp group as we do in these conditions. Yes, did you copy everybody? So what are you? You are holozoic nutrition. You are having holozoic nutrition, isn't it? Holozoic means you are just chewing down your food, breaking down it in complex to simple form in your stomach, in your intestine. We did all those things in your stomach, in your intestine, in your the digestive system, and then you assimilate it or you absorb it. Okay, fine. So let's start it. So similarly what we have done, because this seems to be complex, isn't it? Whatever things I am teaching up till now, this seems to be a bit complex. So what we have done is that, so we'll be dividing the members of animal kingdom into different categories, into different phyla. Dividing the members, or we will be just classifying them in different phyla. What are the phyla? Let's see. First and foremost, we will start up with Porifera. Okay, before you start copying, you need to know that just make your copy horizontal. Just make your copy horizontal so that you get a lot of space on the sides, and write these particular phyla in the center. Okay, for example, if this is your copy, just don't use in this way. Okay, just this is a notepad. You can just use it in this way and write in the center everything, so that you get space in these particular margins. So let's write Porifera. We have Coelenterata. We have Ctenophora, Platyhelminthes, this is Cteno. We have Aschelminthes, we have Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. So you have to learn these 11 phyla in this sequence only, because this is what is the sequence of their phylogeny or their evolution. So you cannot break this sequence. So when you consider Porifera, it needs to be low in evolution compared to Coelenterata. When you consider Aschelminthes, it is higher in evolution compared to the rest of the phyla placed before it, that is Porifera, Coelenterata, Ctenophora, Platyhelminthes. So Aschelminthes is superior to that, and obviously the last phyla, that is our phyla, Chordata, they are the most superior or the highly evolved group of animals which are placed the last. So based on the evolution, we have started from the first and end on the start, just like plant kingdom. You must have done plant kingdom chapter also. So similar to plant kingdom chapter, this will be done over here also, exact same. Now, so moving ahead, what we have next is, we will be discussing about how to classify these 11 phyla. We have written 11 phyla, but there must be a system of classification that must have given us that this is the particular phyla, this is particular organism belonging to this phyla. So let us start with the classification system. So there is basically, what we talk about, first and foremost thing, levels of organization, isn't it? What are the levels of organization? The first thing that gets us to know that yes, what is the basis of classification. So level of organization that comes across to our mind is, first and foremost, the animal might be having just a simple aggregate of tissue, cells. So the first in this list is cellular aggregation or cellular aggregates. So the animals belonging to this particular class, they are just aggregates of cells. Aggregates of cells means some of the cells, they come together and they start performing different similar kinds of functions, but yes, there is a very little division of labor, because division of labor term will come only when you talk about tissues. So they don't have proper division of labor. The only phyla showing this characteristic feature is phylum Porifera. So Porifera is having cellular aggregates. They don't have tissues, they just have many cells grouped together. They don't have tissue, because the definition of tissue is what? Definition of tissue is that a tissue is a group of cells having same origin and same function. So they don't have same origin, but yet they have a basic level of similar kinds of functions, basic. Next, what we talk about after cellular aggregates, number 2 comes across is tissue level organization. So tissue level organization, now they are having groups of tissues, isn't it? Groups of cells have come together and they are having what? They are having similar origin and function. So the phyla which are having the characteristic tissue level organization are the next ones, Coelenterata as well as Ctenophora. Just keep on marking these things, mark in orange. Just if you have multicolored pens, it's good, or just keep on underlining with the same color pen if you have. Now tissues, they will come together to form what? To form organ, isn't it? So now comes the organ level organization. What happens is groups of tissues start to form organs to execute same functions. The examples of the animals having organ level organization are from the only Platyhelminthes. Only Platyhelminthes happens to be the phyla or phylum to have organ level organization. Next comes is organ system level organization. So organ system level organization is what? What are they? Yes, groups of organs execute a same function. For example, stomach is an organ, but alimentary canal is an organ system. So stomach, intestine, buccal cavity, so together they form is alimentary canal. So alimentary canal or GIT, that is gastrointestinal tract, or the digestive system will be an organ system. So from Aschelminthes, let's see. To chordata, all the phyla included in between are having organ system level of organization. Clear? Everybody? Let me know if you have copied. We'll go back to this slide and we will. So what do we have? Porifera. What is Porifera having? Yes, they have cellular aggregates, or just write down cellular because we need to save some space. And then we have is Coelenterata and Ctenophora. They are having tissue level organization. Then we talk about Platyhelminthes. They have organ level organization. From Aschelminthes to chordata, what we have is organ system level of organization. Fine? Clear up to this? Everybody, all the centers, let me know. If it is not, I will try to explain again. Yes, children? Copied? Clear? Shillong, Tamilong, Senapati, Itanagar. Clear with this? Everybody? Okay, very good, Senapati. Rest of the centers. So Ashkelminthus will have an alimentary canal, isn't it? But Platyhelminthes will not have an alimentary canal. So they are said to have organ level organization. Okay. Clear? Okay, children. So let's move to the second level of organization. So under levels of organization, this was the first thing. Now let's talk about next, that is symmetry. So what is symmetry? What is symmetry? Tell me, what is symmetry? Symmetry is the ability to cut a particular thing into two equal parts. Like for example, if I wish to cut this mobile phone into two equal parts, I would be able to, isn't it? From two equal parts, I can cut it. I can cut it this way also, this way also. So this is symmetry. This is showing what? This is showing symmetry. Now, next thing is that when we talk about symmetry, we talk about many more things accordingly. We talk about first and foremost about those animals who don't have symmetry. We'll talk about asymmetrical animals. So asymmetry is something, in either of the ways if you wish to cut, they will not be giving you any kind of equality. Okay, so the only phyla to be asymmetric is Porifera. So what is asymmetry? No symmetry from any plane. Next is talk about radial symmetry. So radial symmetry, you need to think about something which is round in structure and you are cutting it actually in a way that you are getting equal parts. So if I am having a round structure, if I am cutting it into equal parts, yes, I am able to cut. Isn't it? This is what is called as radial symmetry. You just need to think about pizza. Pizza is having radial symmetry. So what is radial symmetry? Radial symmetry is basically kind of symmetry that cuts the, since we are talking about animals, organisms into equal parts. Mind it, it is equal parts. This is radial symmetry, cutting the organism into equal parts. Next comes the bilateral symmetry. What is bilateral symmetry? When you are trying to cut it into two equal parts that are left and right. This would be the left part, this would be the right part. So bilateral symmetry is axis cuts the organism into equal left and right halves, isn't it? So left and right half is of more importance when we talk about what? When we talk about bilateral symmetry, and radial symmetry is equal parts. So the animals having asymmetry is Porifera, radial symmetry is Coelenterata, and Ctenophora and Echinodermata. So we have 3 phyla having radial symmetry. Bilateral, obviously, except Echinodermata from Platyhelminthes to Chordata, but you need to accept the Echinodermata, because yes, Echinodermata is radially symmetrical. Echinodermata is a starfish. So let's see how to write over here for the column. So what do you have? We have this, along with this is asymmetrical. I'm sorry. This is asymmetrical. This is radial. This is radial, but all of them, rest all, from Platyhelminthes to Chordata, all of them are bilateral. But the only exception is what? The only exception is Echinodermata, which is having radial symmetry. Clear up to this? So these are 2 levels of classification. We will talk about cellular aggregates, tissue level, organ level, and organ system level of organizations. Next point, what comes in our mind is symmetry. Symmetry has asymmetry, porifera, radial symmetry, coelenterata, ctenophora, and Echinodermata, and then bilateral symmetry, that is in equal left and right half, Platyhelminthes to phyla Chordata, but not Echinodermata. Clear? Keep on copying this table, updating this table. So have you learned something till now? Are you able to learn something till now? Yes, Shillong, Tamilong. Yes, children. Let me know. Did you learn something? Yes. Tamilong, Shillong, Senapati, Itanagar. Okay, fine. So name the phyla. Let's see a question. Okay, we'll keep on having some questions because I know this chapter is bit. Okay, let's have a question. Name the phyla. Okay, I will be giving you the properties and you have to name the phylum based on that. It can be one phylum or it can be more than one phylum. Okay, let's see. Properties. The first property is, we'll write 1, 2, 3 and we'll have 1, 2, 3 ABC options. The first property is, it is radially symmetrical. You have to guess the name of the phyla. Obviously, not guess, but you have to give me correctly the name of the phyla. Okay, it's radially symmetrical. It is having organ system level of organization. It has no bilateral symmetry, in adults, but has radial symmetry or modify this point in a way it has bilateral symmetry in larvae but radial symmetry in adults. Okay, perfect. So, did you see all the particular statements? Let's name the phyla. What can be the options? C. Porifera. Second option, Coelenterata. Third option, Echinodermata. And fourth option, all of these. Yes. Quickly answer the question. Yes, children. Should not take this long. This is an easy question. Should not take this long. Yes. Shillong, Tamilong, Senapati, Itanagar, please try and answer this question. Good, Shillong. Nice. Shillong boys stood eco, isn't it? I guess there are 2 classes still. In one there were 9 people, 9 children, and the remaining in the other class, isn't it? Yes, Shillong, I'm waiting. Quick. Cannot take this long. Yes, Tamilong, Itanagar, Senapati, please answer this question. Don't sit idle, please answer this question. Very easy question. We are going to have many such questions in your NEET also. Yes, quick. Ijanagar, you people are only left. I have answers. Tamil Long, rethink your answer once again, please. Yes. Yes. Good. Nice, Tamil Long. Yes, Itanagar, you people are only left. Please answer. Please answer this question. Yes, it's C. Accurately, it's C. Very good. So let's see what was the classification system we followed. See, the first thing was it has radial symmetry. If you move back to this table is self-explanatory. You can just have your answers from this table as well. That we talked about radial symmetry. So radial symmetry is present in 3 of the phyla. That is Coelenterata. Radial symmetry is present in Coelenterata, Ctenophora, and Echinodermata. These are 3 phyla which have what? Which have radial symmetry. So you need to check out the phyla which have radial symmetry. Then the next thing was what? It was about Echinodermata. That is the organ system level of organization. If you see the second point, second statement, they were talking about an animal which had organ system level of organization. So the only animal to have organ system of organization as well as radial symmetry comes out to be what? Comes out to be Echinodermata. Then you had an additional information given over here. It was good. You can add this point for your Echinodermata section when we will be moving ahead to each of the phyla, that it has bilateral symmetry in larvae. Okay. The larvae of the Echinodermata, they have exceptionally bilateral symmetry, but yes, the adults, they don't have bilateral, but they possess radial symmetry. So this is a very, very unique feature. You need to add on all these 3 statements are the indicative properties that we are talking about the only one phyla which happens to be Echinodermata. So your option should be C. C is the correct option for this question. That is Echinodermata. Clear? Everybody? Sorted? Any problem? Yes. Ijanagar got the way to solve these kind of questions? Okay. So clear up to this? So let's proceed. So next thing what comes in our mind, isn't it? Next thing what comes in our mind after 2 things, that is first the level of organization, the second we are talking about symmetry, the third what comes in our mind is the number of germ layers. So what is a germ layer? We are dividing the animals on the basis of germ layers. What are germ layers? Tell me. What are germ layers? Any idea? What are germ layers? Yes. Quickly, Shillong, Tamilong, Senapati, Itanagar. What are germ layers? Tell me, children. Yes. Okay. What are germ layers? You are consulting and then rushing towards the keyboard. Okay, fine. What are germ layers? Yes, quickly. Good. Very good. Good, Shillong. So, on the basis of the number of germ layers, that is how many germ layers, it's ectoderm, not exoderm, okay? ECTO. So, on the number of the particular germ layers, yes, on the number of germ layers, we can have 2 category of animals, 2 category of organisms. The first is said to be diploblastic and second is said to be triploblastic. So what are germ layers? Germ layers are the particular layers which gives rise to tissues or organs of the organism. Okay. So a diplo, let's see which are the layers which are present in a diploblastic animal. So let's so this is the ectoderm, this is the mesoglia, not mesoderm, it is mesoglia, and this is endoderm. What is mesoglia? If a question comes to you, you need to answer that this is a non-cellular gel-like layer. It is non-cellular gel-like layer, and this is endoderm, and this innermost layer happens to be gut. Oh, better I don't color this because not able to follow the drawing rules. So this is the particular layer called as diploblastic, particular structure called as diploblastic. Now when you refer to the triploblastics, let's see quickly what do they have. So this is the outermost layer called as ectoderm. This is the innermost gut. This is the middle one. Now the middle one is called as mesoderm. And this is the innermost, again called as endoderm. So we have what? We have a different layer, different structure over here. And what is that structure? We have mesoglia, and over here the mesoglia has turned into mesoderm. Obviously the mesoderm has a different characteristic property, that what? It has now become cellular. So this non-cellular layer is just like a gel-like structure which is responsible for giving them compaction, just making the layers compact. Okay, so this has not much function, but it functions to give compactness to the whole structure. This is gut. So we have three layers. What are they? That are endoderm, mesoglia, ectoderm, where the mesoglia is non-cellular gel-like, and three layers in triploblastic, that is ectoderm, mesoderm and endoderm, where the mesoderm is very much cellular like any other layers like ectoderm and endoderm. So what is basically the role of this three-layered structure, two-layered structure? See, what people think is that there are only two phyla. Okay, there are only two phyla that are basically, they are called as Coelenterata and Ctenophora. Two of the only phyla. Okay, we will not talk about Porifera, since Porifera is just aggregates of cells. We will not talk about Porifera, but we will talk about Coelenterata, Ctenophora. Yes, Coelenterata and Ctenophora, they have diploblastic level of organization. Why? Because they have certain tissues. Till now the organ formation has not taken place. The organ formation is not started. So if you go back and see over here on the particular slide, the 7th slide, you will be able to see that organs, they have started to form from platyhelminthes onwards. Okay, so obviously there must be a kind of interaction, isn't it? There must be a kind of interaction that is present from platyhelminthes down to chordata. from platyhelminthes to chordata, there must be a particular interaction between what? See, these all have organ and formation of organs have started, organ system has started. So, we will talk about something special over here, that is mention interaction of mesoderm with endoderm is essential for mesoderm with endoderm is essential for what? Organ formation. So you can correlate the two things, that yes, from where we have started triploblastic, from there only you have to start organ level. So you need to remember this thing also, this is a key point. You have to remember this thing, that triploblastic and organ level organization will start from the same point. Where? From the platyhelminthes, because the reason being that the mesoderm and endoderm should interact with each other to form an organ. So till the mesoderm has not been formed, we will not have any organ, and that is what we have seen over here also. If you refer to the particular chart that I asked you to make in the center of the paper, so this is what is basically. So we can just add over here one more point, that yes, they are diploblastic as well. So Coelenterata ctenophora, they have three properties till now. What are they? Tissue level organization, radial symmetry, and they are diploblastic. When we talk about rest of them, each one of them from platyhelminthes to last, just mark it down, from platyhelminthes to the last chordata, the particular characteristic is what? That all of them are having triploblastic organization in their germ layers. All of them are triploblastic. This is diploblastic. Clear with everybody? All of you are clear with this? Do you mark everything on your paper, on the particular sheet? Okay. Let me know if anything is not clear. Shillong, just give me thumbs up if things are clear. Yes. And the other section, let me know if it is clear. Yes. Shillong, please. Okay. Yes. Okay, very good. Senapati, Tamilong, Itanagar, Shillong. Are things clear? Okay, very good. Okay. Nice. Done? It's clear? Tamilong, clear? Shillong, clear? Copied? Copied everything? Okay. So we have completed with three things. That is, first was what? First was the level of organization, whether it was cellular, tissue, organ or organ system. Then we started with the particular symmetry level. The symmetry, that is whether it is asymmetry, radial symmetry or it is having bilateral symmetry. Then we talked about germ layers, that is the organism, whether it has two layers, in between there will be a mesoglia, non-cellular, or it has three layers, mesoglia converted into mesoderm, that is triploblastic. Okay, fine. So three things are left, three more things are left. We will have a small break, 5 minutes. Let's have a small break of 5 minutes, and then we will discuss about the three remaining things. Okay, fine, no doubts. Just go through it in the meantime. Let me know if there is any problem. Okay, just 5 minutes. 5 minutes of break. Okay children, let's resume. Back from the break, everybody? Yes. Is everybody back from the break? On the class? Okay, fine. So let's proceed. So before we talk about the next level of organization, level of classification, I'll just give you a brief idea that why was these all things done. See, the first and foremost thing what started to have give an idea about cellular organization is that these cells, they are aggregates. Okay, just we have started making our cells from unicellular to multicellular. So just now I told you that protozoa is said to be the little animalcule, isn't it? Protozoans are said to be little animalcules, and they have started to form aggregates, cellular aggregates. So there is something which is basically just started, and we have placed that all those animals which just started aggregation of its cells in the particular phylum called as Porifera. Now next level of cellular aggregation would be what? There will be need of tissue, need of a particular level of division of labor. There should be division of labor so that every cell has to participate in a single function, not multiple functions. That will be giving them more of differentiation. So the accuracy level increased and they started to follow tissue level organization. Same pattern followed up organ level and the last organ system level. Now once organ systems were formed, what was the next need? The next need was a symmetry. Now how is symmetry associated? Now once the animals were having cellular aggregates, there was no orientation. They don't have any orientation. They are just as such mass of cells. Now radial symmetry is a bit of more evolved form when you compare it with asymmetry, in a way that now the orientation has started to form. It has just started to form, but it has not completed. But when you talk about bilateral symmetry gives a particular level of organization in a way that animal has formed a head. All the organisms which are having bilateral symmetry, they have cephalization, that is formation of head. And their body is perfect now for the activities of different kinds like predation, escape from the prey. So this bilateral symmetry gives them more of organs. Organs can be accommodated in a proportion. That is what is the advantage of bilateral symmetry. Then we have the layers, isn't it? Layers. The diploblastic, triploblastic, all those layers. Obviously, if I am talking about a particular layer, that is going to give rise to the next tissues, the next organs. So if you are talking about diploblastic, obviously diploblastic is the layer which is having only two of the germ layers, and the germ layers will be what? Ectoderm and endoderm. Now when mesoderm starts to come, because what is the need of the hour? Now we need is more evolution. Platyhelminthes needs more evolution compared to the previous phyla, that is Ctenophora. So what Platyhelminthes will do? Platyhelminthes will start to form a new layer, that is mesoderm, and that mesoderm gives what? The mesoderm starts to give them the appearance of alimentary canal. The alimentary canal has started. It will not be complete because we don't talk about organ system level in Platyhelminthes, we talk about only organ level organization in Platyhelminthes. So it has started to form, but it will not be complete. Now next thing what will be? The next thing will be to have an idea that yes, my organs are having sufficient of space to be placed. Now based on that category, I have coelom. The next division will be on the basis of coelom. What is coelom? Coelom is the body cavity between gut and body wall. I'll just send you an image. You can just, to accommodate, you can just refer to that. Accommodate different organs. Like if talk about kidney, your kidneys are placed in coelom. If you talk about your heart, your lungs, your pancreas, your liver, all these organs are placed in your coelom. So coelom is what? Coelom is a particular structure that gives an additional space to accommodate many of the organs that are not present in those animals which don't have a coelom. Let's quickly refer to this. Just send you the image. Just let me know if you can refer to it. Yes. Can you see this, everybody? Can you see coelom? You must be able to see coelom now. So coelom is a particular space or body cavity where you have inside gut, remember gut, just now we did gut, and your body wall. Body wall means the outer surface of your body. This is body wall. So in between the particular layers you have many organs. You have stomach, you have liver, you have kidneys, you have pancreas, you have a large, very large area of your small and large intestine. So that all is courtesy coelom. So gradually as the evolution started, the organisms, they started to have a growth in their body, isn't it? A growth in their body means the organs additionally need an extra space. The intestine needs to be longer, because if the intestine is not longer, the animal will have a tendency to quickly feed. The feeding time will be decreased. So if feeding time is decreased, frequent exposure to the outer world will obviously invite more predators. So what animals they started to? The animals they started to have a longer intestine, so that food can be accommodated, food can be kept in that intestine for a longer duration of time. Isn't it? We have studied that in digestion also, that for example in humans 4 to 5 hours is like the stomach is keeping your food, and then the digestion process proceeds and it moves it ahead to the intestine section. So gradually what happens? Gradually we are talking down about the increase in evolution, the increase in the characteristics of an animal. So the coelom is what? What is a coelom? Coelom is a body cavity between gut and body wall to accommodate. The first thing for coelom would be what? The animals which are triploblastic, they are only having this qualification to have coelom. Before triploblastic, we will not talk about coelom only. We are not going to talk about coelom in porifera, not in coelenterata, not in ctenophora, because yes, coelom is what? Coelom is a body cavity to accommodate different organs. So organ formation started from where? Organ formation started from platyhelminthes. So you will talk about coelom if present or not from platyhelminthes to chordata. If the 3 layers are not present, there is no organ. No organ means no coelom, because if organs are not there, why would be the coelom present to accommodate the organs? Am I clear to everybody? So what is the first thing? Is the first thing we talk about? Sorry. And talk about those animals who are coelomates. So coelomates, it's very simple. What you need to do is that you just need to draw the 3 germ layers, that is ectoderm, mesoderm, not glia, and endoderm, and the innermost obviously gut. So acoelomates, they don't have any body cavity. A means absent, coelomate, that is coelom is present or not. The only phyla to have no coelom is Platyhelminthes. So the only phyla that is not having any coelom is Platyhelminthes. They don't have a coelom. Clear? Next we will talk about, so there are three animals actually, three categories. One is pseudocoelomates. Pseudo means false. Okay. So just talking about an instance, and we have coelomates. So coelomates means having true coelom. And what is pseudocoelomate? They have coelom but it is false, and they are true coelomates. Okay. So first, to know what is false, we should know what is true, isn't it? To know the truth, you should first, to know the lie, to know a lie, you should know that whether this person is speaking lie or not. You should be able to judge only when, you will be able to judge only when you are aware about the truth, isn't it? So you need to talk about truth first. We'll see truth first. Truth, let's see what is truth. A bit of shaky diagram. Let's see quickly. This is the ectoderm. Okay, so just taking down this color. Just ignore my blunders if I am not putting them well inside the lines. It's actually a blunder in painting. Okay, so this blue section is what? First I need to mark this blue section in this thing. Okay. So if you wish to refer, this blue section is mesoderm. Now, can you see in between the mesoderm a cavity? Can you see in between the mesoderm a cavity of yellow color? You must be able to see. This yellow color is nothing but it is the coelom, the particular space that has been created by a true coelomate to accommodate its organs. This is mesoderm, inside and outside, and this happens to be what? This happens to be coelom. And this is ectoderm, and the innermost layer is, as we are aware about it, endoderm, and this is gut. So So these are three typical layers which are oriented in a way to form the coelom. So a true coelom is a coelom which is always lined by mesoderm from inside as well as outside. So true coelom is what? Is lined by mesoderm just a moment. Yes. Oh, this all disappeared. Sad. Okay, fine. Quickly, let's go ahead and draw this all. Okay, I need to draw this again. There was some technical error. So let's move ahead. Yes. And we selected yellow and we mark this along. Okay, fine. Done. So this was mesoderm. This was also mesoderm. This was coelom. This was endoderm, and this section was what? This was gut. Clear? So this diagram is clear to one and all. Everybody? Yes, children. Shillong, Itanagar, Senapati, Tamilnad. It's clear? Okay. Now you know the truth. What? That true coelom is lined by mesoderm. Always. Outside, inside. Both the sides. On both the sides. It's lined by coelom. Lined by mesoderm. True coelom. Now let's see what happens with a pseudocoelomate animal. Now this is ectoderm. So can you see the difference? Now what is happening is mesoderm is actually present inside the coelom. Coelom is lined by mesoderm, but in a different way. This is what is a pseudocoelomate. So this blue section, as we have marked from the beginning, is mesoderm, which is in the form of pouches. Patches. This is the endoderm and this is gut. So you have this particular kind of orientation in a pseudocoelomate animal, that is the particular layers, that is mesoderm, is actually lined by coelom from everywhere. The only phylum which is, the only phylum which is pseudocoelomate happens to be Aschelminthes. Only phylum which is pseudocoelomate is Aschelminthes, and the rest of the phyla, that is from the Annelida to Chordata, without any exception, is having true coelom. Once again, coelomates, the animals which have a body cavity to accommodate the extra organs, icoelomate Platyhelminthes, pseudocoelomate Aschelminthes, and then we have coelomates from Annelida to Chordata. Let's mark down over here in the table that we were making. Yes, this thing. So we will not talk about coelom in the Porifera, Coelentata, Ctenophora. Why? Because they don't have the layers. So you need to have mesoderm first of all. Mesoderm is present in which of the animals? It is present in Platyhelminthes, but yes, this is a coelomate. It is present in Aschelminthes. It is pseudocoelomate. But when we talk about the animals from Annelida to Chordata, they are what? They are very much coelomate. So we have this particular criteria, this basic division that is taking place when you talk about the coelom, when you talk about the layers, all those things. Clear to everybody? Just let me know if it is clear. All of you. Itanagar, Shillong, Senapati. Is it clear? Okay. Shillong, is it clear? So are you getting something? Are you able to learn few of the things? Okay, you should be able to learn also together with that, along with that. Okay. So next is what? Next is the level of organization we will talk about is segmentation. Now segmentation is what? Segmentation is that the body is divided into metamers or in segments. Now the advantage of having segments is that each segment has its own blood vessels, its own muscles, its own nerve cells, and sometimes, not always, in some cases, reproductive organs also. Each segment. So this is said to be what? This is said to be segmentation. So segmentation is what? The body is divided into metamers or segments. So this division is only present in only 3 of the phyla. They happen to be first Annelida, the second happens to be the Arthropoda, and the third happens to be Chordata. So we have only 3 phyla showing this characteristic property of segmentation. Let's star mark in this also. It was, I guess, slide number 7. Yes. So let's quickly mark down in this also. So we have Annelida, we have Arthropoda, and we have Chordata. So just tick marks. What they show is, the tick marks show the segmentation. So the phyla with double tick marks, they are the ones which are segmented. You can just mark it with other shade also if you wish. Let's mark them in sky blue since I have lots of colors. So just okay. The blue marked ones, they are the ones which have segmentation. Clear? Done with this? Everybody? Okay. So let's move to the last slide once again and talk about the last classification system according to your NCERT. So your NCERT tells that there is a particular presence or absence of sixth structure called as notochord. So what is notochord? Notochord is a rod-like structure present on the dorsal part, dorsally present, derived from mesoderm. So it's said to be notochord. There is only one phylum without any exception, that is the Chordata, which has notochord. Only one phylum. So rest of all, the 10, they are non-chordates. Only chordates are having notochord. So this was in this slide. That's all. So this is all about your NCERT. That is what is the books they tell you. I will be adding one more thing on the division level, that is the appearance of a particular structure. On that basis, we have two kinds of organisms. One is said to be protostome and one is said to be the deuterostome. Okay, so protostome is the one where the blastopore is the particular structure that is present in the blastula. Okay, just a small pore-like structure. If the blastopore develops into mouth, the animals are said to be protostome. But if the blastospore develops into anus first, if the blastopore develops into anus first, it's said to be deuterostome. So you have all the phyla, okay, every phyla, all the phyla from Porifera to Mollusca. From Porifera to Mollusca, all of them are having development of mouth first, that is they are protostome. And the only phyla, Echinodermata and Chordata, they have development of anus first. So deuterostome is said to be evolutionarily more advanced compared to protostome. Deuterostome is more advanced, protostome is less advanced. So based on this character, we have placed Echinodermata in the last. You must be wondering that if it is having radial symmetry, why we are placing it downwards, isn't it? This is the reason why we are placing it downwards, because yes, it might be having radial symmetry, but in no ways the deuterostome section is more stronger, that is the development of anus takes place first, and that makes it more evolved compared to the rest of the phyla. See, So you will be placing this. I am talking about. We are placing anadromata in the last, isn't it? We are placing it over here. It is having radial symmetry, but you are placing it over here. That clearly marks that it must be evolutionarily somewhat more advanced, isn't it? So we are having echinodermata and chordata. They are the ones which are deuterostome. Rest all are protostome. Clear? Everybody? So let me know if you have copied everything. Everything is clear. This is all about the level of organization. Just let me know if it is clear up to this. Yes, children. Shillong, Tamilong, Senapati. Just copy this down, please. If you are missing out on something, yes, I'm here. Don't worry. Yes. Everything, did you copy everything? Itanagar, Senapati, Shillong, Tamilong. Okay, great. This thing, just copy it down if you are missing out. Yes, Senapati, Shillong. Everything is clear? All the centers. Is everything clear? Okay, very good Itanagar, very good Tamilong. Shillong, what about you people? What about Senapati? Yes, Shillong, tell me if everything is clear. Answer me first, then you can pack your bags. Shillong Center, to hurry to leave. Answer me first if everything is clear. Yes, children. You can just give me a thumbs up also. If everything is like we do always. Okay, fine. Okay, copied everything? Not missing out on anything? Okay, fine, very good. So let's we will be starting from porifera in tomorrow's class. Okay, fine, very good. All of you, very good. So we will start with porifera in tomorrow's class. Let's see how many phyla we will complete. I guess we should be able to complete this whole animal kingdom in 2 classes. But let's see. Understanding is more important rather than rushing through a topic. But yes, remember to have your NCERT book along with you when you are studying this chapter, because this chapter is very, very important. Each and everything is always and always coming from NCERT. Okay, I will be giving you some tricks how to learn the names and examples tomorrow. Okay, fine. So everybody is in hurry. So let's meet tomorrow. Okay, bye bye. Thank you so much. Good evening.